It's a great pleasure for me to be here, and it's a real honor, in fact, because it's cool. It's really cool to have the opportunity to discuss about the results we recently obtained in my small research group in Dijon. And this talk will be dedicated to the existence, prevalence, and functional relevance of alternative nucleic acid structures. And I decided, because I discussed with Valérie and Emeline, to provide you with some kind of historical background regarding the alternative nucleic acid structure, just in order to provide you with the keys to better understand what we are currently working on and why we are currently working on that topic. So it will be a bit maybe too superficial for the people who are already involved in the G4 field, but I think that everybody, the take-home message will be at least crystal clear, I hope so. So so you see my pointer, I guess, you see my presentation, I think it's okay as well. So when we discuss about nucleic acid, of course, the first image that pops up into our mind is this wonderful double helix duplex DNA structure that was elucidated in 1953 by Watson, Crick, Wilkins, and Franklin. And this discovery is certainly one of the most important discoveries in the whole history of science for many different reasons. For example, because it provided the support of genetic information, and provide us with the key to understand how cells divide, what are the laws of inheritance, and so on. So this is a very wonderful discovery, and this duplex structure is absolutely well known. But what is less known is the fact that nucleic acid can adopt as well a series of other structures, which are called the higher order nucleic acid structures, or the alternative nucleic acid structures, and can be of 2 strands, 3 strands, or 4 strands structures. For example, here you have the wonderful bDNA. So for the short history, it is called bDNA. This is a Watson-Crick duplex here, because when Franklin resolved that crystal structure would provide the basis for elucidating the duplex structure, she was working in the Wilkins lab, and they received from Switzerland 2 samples, the sample A and the sample B, and Wilkins decided to work on the sample A, and Franklin decided to work on sample B, and finally, the structure that you have here, this duplex, the Watson-Crick duplex, is in fact the sample B. This is what it is called, the bDNA, or the duplex DNA. But some other duplex exist. For example, this duplex of inverted helicity, which is called Z, so it's not related to the fact that the sample is sample Z. It was just because it is weirdly shaped duplex, and Z means zigzag. But you can also have 3-stranded structure, so you just have an additional strand here, the green one, and you have the triplex, or this Y-shaped DNA, which is called the 3-way DNA junction, and you can still have 1 additional strand, for example, here the blue one, and you have the quadruplex DNA for the 4-stranded structures, and this H-type 4-way DNA junction. And clearly, we can see that the biophysicists are not that original, because in fact, this is duplex, triplex, and quadruplex. That is straightforward this way. To explain the existence of this alternative nucleic acid structure is just a simple question of supramolecular chemistry. For example, in the duplex DNA, the 2 strands are held together through a network of hydrogen bonds, or H bond, between the so-called Watson-Crick base pair. The guanine with the cytosine, the thymine with the adenine. You have 3 H bonds between the G and the C, and 2 H bonds between the A and the T. And clearly we see here that this base pairing does not fully exploit the H bond donating and or accepting properties of the nucleobases. For example, in the guanine, indicator guanine, or indicator of the adenine, H bond can be created through this nitrogen, for example, and you can have access to a triplet of base, and this is the basic building block of triplexes. And in the particular case of the guanine, you can have access to the quartet of base, and this is the basic building block of quadruplexes. What is quite fun is the fact that the existence of this higher order nucleic acid structures was done quite rapidly after the discovery of the duplex. For example, the duplex was elucidated, the structure of the duplex was elucidated in 1953, but the triplex was elucidated in 57, and the basic building block for the quadruplex in 62, meaning that 10 years just after the discovery of the duplex, we already knew that some alternative structure can exist, but this was more or less forgotten, mainly because of this absolutely visionary lecture provided by Francis Crick in 57. He was not yet awarded the Nobel Prize, in which he explained on the basis of the structures of the duplex, but also on the wealth of bibliography related to the mysteries of cellular functions. He explained what he has called the central dogma of biology, which just explained the flow of information between the DNA and the RNA, which is known now as the transcription, and then the RNA to the protein, which is now known as translation. And this was a theoretical lecture, but finally 10 or 20, 30 years of research were needed to show that, in fact, this is absolutely the case. So a huge, massive effort was invested in deciphering or assessing the reliability of the central dogma, and it took a lot of energy from the community. And this explained why, even if the existence of these alternative nucleic acid structures was demonstrated many, many years ago, finally, they were considered as lab curiosity for something like 4 decades. More recently, you have even more complex structures. For example, the R loop are currently widely studied. This is some kind of alternative nucleic acid structures, but you have a mixture of DNA and RNA. And also the I motif is quite popular nowadays, and it's a kind of a 4-stranded structure, but the structure do not rely on the formations of guanine quartet. It just rely on the intercalation of cytosine base pair. And because this is intercalated, this structure is called the I motif. Even if this structure looks very different from each other, they share something is common in the fact that they can fold during the same cellular events, meaning that when DNA is at work. So the transcription, the cell division, so the replication, and all of this is known as DNA transactions. And the DNA transaction, for example, you have the example here of the replication, for example, provide a window of opportunity for this structure to fold as a result of 2 different features. The first one is that the DNA, the duplex is separated, so the single-stranded DNA is freed from its duplex constraint, and then the DNA can fold upon himself to adopt, for example, the quadruplex structure, which can be considered as a simple molecular knot of just DNA fold upon himself. But the second feature is the fact that the motion of the polymerases, which are in charge of the DNA transaction along the duplex DNA, create a kind of super helicity ahead of the replication fork. And the way to relieve that torsional stress is, for the DNA, is to adopt a series of alternative nucleic acid structures. For example, the 4-way DNA junction here, also known as a cruciform, or the 3-way DNA junction here, also known as slippage loop. And only the exact sequence of the sequence involved will dictate the type of structure that would be adopted by this sequence. So my lab is here. We are currently working on 2 different structures, the quadruplex and the 3-way DNA junction. Today, I will focus only on quadruplex DNA, but my personal opinion is the fact that the 3-way DNA junction are quite fascinating target for establishing an anti-cancer strategy. We are currently developing this project with Anton Granzen in Curie, in Orsay, and Nicolas Chéron at the ENS Paris, and also with Sébastien Briton in Toulouse. But today I will really focus on quadruplexes. And this is the most important slide, the acknowledgement slide. So this is the members of my group currently. Francesco just defended his PhD, so he's back in Italy. Jeremy is working on a project I won't have the time to discuss about today, but this is a fascinating project in which we are looking for molecules that do not stabilize, but destabilize quadruplexes. So maybe I will have a couple of minutes at the very end of the presentation to explain you the interest of discovering such molecules. And he is working on the three-way DNA junction, and Mark and Sandy are the most involved person in the G4 field. And my collaborator, of course, we will see the name of Judy in Vancouver, of Wenli in China, Andre in the US, and here my collaborator here in Dijon, Ibai Valverde, was a CNRS researcher as well. So quadruplexes. Question, what is a quadruplex? So if you have a DNA strand here, I represent, schematically speaking, the guanine in the gray square. If this DNA strand is full of guanine, it can fold upon himself to create this kind of molecular knot, which is known as a quadruplex, because there are 4 strands. And the stability of this quadruplex is provided by the interaction of 4 guanine in the same plane to form a G-quartet. In fact, it is quite fun to notice that quite rapidly, something like 30 years ago, chemical biologists already designed some molecules to interact with the quadruplexes. Why it is fun is because, in fact, we even didn't know at that time if quadruplex could form in cells, but we decided anyway to target them with small molecules because it was a quite interesting chemical biology quest. So a lot of myriad of examples exist of molecules that stabilize quadruplexes. And this approach has provided a wealth of data, which is so huge that from time to time, it's even very complicated for us to discern what is wrong and what is right and what is the point of this study. And I'm used to summarize this saying that the stabilizations of quadruplex within cells using small molecules, which are called ligands, tend to raise more questions than answer. And today we will try to answer some of these questions. And to this end, I would like to provide you with some historical background regarding the G4. So, of course, everything started with the work of Frédéric Michel quite a long time ago, when he was the first to isolate the substance that is within the nucleus. So he called this a nuclein, and the term nucleic acid was coined 20 or 30 years after that. Hammerstein, a couple of years after, isolated, so did what the biochemists do the best, meaning that they cut everything into pieces and try to understand the basic building block of this nuclein. And he discovered that the nuclein is really rich in the guaninic acid. So this is the basic, I would say, the starting point for the discovery of the quadruplexes, because this, let's say this fundamental unit was isolated, but we didn't know exactly what it is. So in 1910, Bang and Levin tried to crystallize, because this was more or less the only way to have access to the structures of guanine. They tried to crystallize some highly concentrated solution of guaninic acid, and they failed. They failed because they made this observation that each time this guaninic acid solution is too concentrated, then it can form a kind of semi-liquid jelly that it is indicated here. Now we call this an hydrogel. And this hydrogel was absolutely reluctant to crystallization, so they were not able to obtain the precise structures of this guaninic acid, but this was considered as the first insight into the way the guanine can self-interact to form something like hydrogels. The solid structure was finally solved in 1962 by Davis, So there was also some kind of UV investigation performed in Corona as well in 62. But we obtained the firm demonstration that guanine can self-assemble and form a quartet, and then the quartet stack upon each other to form some columnar gels. So the basic building blocks of the quadruplex was discovered in 62. And then I would like to remember that here, this is isolated guanine. So in 88 and 99, a series of Nobel Prizes decided to investigate whether this self-assembly of guanine could also occur in more biologically relevant sequences. So instead of using isolated guanine, they included the guanine within biologically relevant sequence and try to see whether this sequence can fold upon itself to form a quadruplex. And this was the case, more or less. It was a gel study, but it was rather convincing results. And the first solution structure was provided in 92, 2 different NMR of 2 biologically relevant sequences, and we obtained very beautiful structures of the stacking of the self-stacking of quartets. So this is a quadruplex. In 92 as well, which obtained crystal structures of a quadruplex, but the well-known, the most known crystal structures of quadruplexes was obtained in 2002 by Parkinson and Needle, and this is a fully parallel quadruplex, which is the iconic image of the G4 field. In 1997, so Needle and Early described the first molecule to interact with quadruplex, and the idea was that quadruplex can form in the telomeric sequence because telomeres are rich in guanine, and the guanine are not randomly distributed in the sequence. These are blocks of 3 guanine, so this sequence is highly prone to fold upon itself to form a quadruplex. And what is interesting with the telomeric sequence is that it is the substrate of the telomerase enzymes. So the telomerase was quite popular at that time because it was demonstrated that the telomerase is not active in the normal somatic cells, but is highly active in roughly 80% of the cancers. So she's responsible for this, for the oncogenesis process. So all molecule that is able to inhibit the action of telomerase could be considered as a very specific anti-cancer agent. And the idea of Early and Needle was to sequester the telomerase substrate under a form, a shape that is not recognized by the enzyme. So it was a beautiful example of indirect inhibition of the telomerase by sequestration of its substrate under a weird shape. So it works quite well in vitro. But it was 1997. This molecule, this acridone, is considered this molecule is considered as the very first example of the G4 ligands. In 2001, there was a NMR structures of a ligand interacting with the G4 provided by SER. It was followed a couple of years after by the first crystal structures of a molecule interacting with the G-quadruplex, and it confirmed the fact that the molecules that are usually flat and aromatic molecules literally sat atop the accessible G-quartet of a quadruplex. In 2002, Early provided the first demonstration that when a quadruplex is not necessarily located in the telomeres, but they can be located also in promoter regions of several genes. And with a small molecule, if you are able to stabilize this promoter that is present in the promoters of several genes, in that case, the MYC genes, then you can down-regulate the expression of that genes. So it was interesting because it was the first insights into the fact that G4s are not necessarily and only restricted to the telomere regions, and they can be also elsewhere in the genome. But the actual question was, in fact, the quadruplexes are certainly an interesting target, but do they fold within cells? Because it's not easy to demonstrate this. So there was 2 demonstrations provided by the group of Shankar Balasubramanian in Cambridge in 2012 and 13. I really appreciate this because this study is absolutely beautiful. They used a clickable ligand, this PDS alpha here, and they incubate live cell with the PDS, they fix everything, and they just perform a step of in situ click chemistry to label the G4 ligands once in interaction with its target within cells with a fluorophore to make it detectable, and then it works quite well. And these results were further supported in 2013 when Shankar published the BG4 antibody, with the well-known BG4 antibody, and he was able to provide some immunodetection of G4 in cells. So this was the two first demonstrations of the existence of G4 in cells. And then from that point, everything went faster. Shankar still developed two different types of sequencing-based technologies, the G4-seq and the G4-ChIP-seq, to demonstrate that G4 can actually form in a genome-wide scale, so in vitro for the G4-seq and the so-called in vivo for the G4-ChIP-seq. So we will discuss about this result a bit later. And in 2020, finally, Shankar was able to demonstrate using this SAR-pi PDS that G4 can actually fold within living cells. So these are the most, to my opinion at least, are the most important dates of the history of G4. But in fact, what is true for the DNA can be also true for the RNA, meaning that a guanine-rich RNA sequence can fold upon themselves to form a G4 as well. And this is exactly the case, because if I look at this timeline, the first investigation by Bang, Levin, and Davis were dedicated to the use of RNA and not DNA. So the story can be repeated again. I will go a bit faster with the RNA. So the starting point is always the same, 10 for Bang and Levin and 62 Corona and Davis. But in 1991, Moore provided the first demonstration by gel that guanine-rich sequence, but this time RNA sequence, can actually fold into quadruplexes. This was tetrameric quadruplexes. In 1992, he provided the demonstration by NMR that tetramolecular RNA quadruplexes can fold. And in 2001, Deng and Xiong reported on the crystal structures. I think it's the very first crystal structures of RNA quadruplexes. Then, 2007, still the group of Shankar provided the first demonstration that a quadruplex within a sequence, an RNA sequence, can modulate the expression of the gel, the gene. So it's just like the demonstration provided by early in 2002. The quadruplex was in the promoter region of a gene. In the case of RNA, this quadruplex is in the 5' untranslated region, so 5' UTR region before an open reading frame that will encode the luciferase, for example. And then what Shankar showed is that if this quadruplex within this 5' UTR is folded, then the luciferase expression decreases. So again, you can modulate the expression of several genes or transcripts using the quadruplexes. It did this without a ligand, but in 2010, it did this with a ligand, which is called the R110. This is Rafael Rodriguez's work. And it works quite well. And then our contribution was to show in 2015 that quadruplex RNA can fold within living cells. We will discuss about this a bit later in this presentation. And then Shankar and Bartel disagree on the existence of on the G4, on the RNA G4. This is also called the RG4, using several sequencing-based technique, but the RG4-seq results are rather convincing. But again, we will discuss about this a bit later. And finally, our contribution was to resolve this conundrum, because the conclusion of Bartel was that RNA G4 do not fold within cells, within living cells. And the conclusion of Shankar was, no, in fact, they do fold, and they fold abundantly in living cells, but they disagree on that point. So we developed a technique which is called the GARP-seq, or the G4-RIP-seq, demonstrate that, yes, G4 can fold within living cells. But again, we will discuss about this a bit later. So this is the whole story of the quadruplexes. And if I have to sum up just in a very brief manner, I will say that, to sum up, quadruplex are absolutely everywhere. In terms of DNA, it is now currently considered that, at least demonstrated in vitro, that more than 800,000 sequences can fold a quadruplex within our genome. And using in silico investigation approach, it is considered that more than 1.5 million sequences do exist within our genome. So it's huge. For the RNA, it's more or less the same. So Shankar demonstrated that more than 10,000 sequences were identified in a set of purified messenger RNA. And Jean-Pierre Perrault showed that there are more than 1 million sequences that are predicted to adopt a G4 within RNA sequence. And it is currently considered that more than half of the human transcript has at least one G4 within its sequence. So again, this is huge. So if this is the actual question of that field, the actual problem of that field, if quadruplexes represent a novel level of regulation of gene expression, either at the transcriptional level for quadruplex DNA or at the translational level for the G4 RNA, is it wise to use a small molecule to control this event? It is appealing. I mean, from a strategic point of view, it is really interesting, but the task might be far too complicated because, in fact, you have here a series of molecules that have been published over the past years. These are very efficient quadruplex ligand in vitro, but the fact is, you clearly see that they share something in common to be flat aromatic molecule that will interact with G4 by stacking atop the G quadruplexes, meaning that why these molecules are very interesting tool, they fade in being considered as therapeutic agent for the reason that they interact with the G quartet, meaning that the G quartet of every quadruplex is exactly the same. And the structural difference between a quartet of DNA and a quartet of RNA is not that huge, meaning that molecule will interact with many different quadruplexes. We do not have at present a molecule that is able to recognize a single G4. And when I speak about G4, is the fact a single G4 is the fact that all sequences are different, so all the folding pattern is different. So a lot of G4s are unique. And for the moment, a small molecule, for example, the FINDC3, will interact with every G4 in the same manner, so with the same efficiency. So it will not be able to discriminate between different G4s. This is a problem and the main challenge of the field. And another challenge is the fact that we do not have in hands a molecule that is able to recognize, to discriminate at least DNA and RNA, which will be the first entry towards specificity. But for the moment, some prototypes are quite okay, but the demonstration of their actual ability to discriminate between two within cells is not that convincing. So this is the major challenge. So, but in fact, if you look at the literature, you will find many different examples of the use of G4 ligand for therapeutic approach to fight against cancer and so on. So another way to look at the story is the fact that, as I told you, DNA transaction provide a window of opportunity for G-rich sequence to fold into a G4 structure. So one way to explain all these results, and this is my personal opinion, is the fact that, in fact, if you stabilize a G4 with a small molecule, whatever the G4, you will slow down the motions of the polymerases in charge of the DNA transaction, and this is recognized as a DNA damage. So finally, it is really interesting to analyze this wealth of data that is available regarding the targeting of G4 using small molecule through this perspective of DNA damage. And I summarize here a series of very convincing studies, start to be quite old for some of them. But the work of Jean-François, for example, was already published in 2006, so it was a pioneer research. if I look at these different molecules, the statin, the 3,6-OA, the PDS or the CX, for example, these studies clearly demonstrate that these molecules are able to create DNA damage in a structure-specific manner. So it's really good. I mean, it's a beautiful way to fight against cancer cell proliferation, but we are far from the fact that the ideal fight of the fact that I stabilize the G4 and I don't regulate the expression of, no, in fact, I just stabilize the G4 and then create DNA damage. And this DNA damage are structure specific, so maybe more cell specific than anticipated. And what is interesting, final point, is the fact that if this molecule are able to create DNA damage, so it can be used as therapeutic agents, they can be included as well in a synthetic lethality strategy. So in fact, you combine different therapeutics, the G4 ligand to create DNA damage, but also the inhibitors of DNA repair, and you obtain a very effective anti-proliferative response. There are several types of synthetic lethality combination that were described, and all of them are very, very active. Anyway, this was my very, very long introduction. I just have a look to the, yes, for the time, it's okay. So what are contribution here? Several years ago, we started roughly 10 years ago, we developed a series of molecules that were absolutely different from the G4 ligand, and just present you because the G4 ligand, most if not all G4 ligand that are currently used in the literature are flat aromatic molecule to stack on the G-quadruplex using the accessible G-quadruplex. We designed in 2011, so it's quite old right now, a molecule which is called TASC, or template assembled synthetic G-quartet. The idea is that we just gather 4 guanines here in pink within a single scaffold. And this molecule exists under 2 conformations, an open one in which the guanine are free to move, and an open and a closed one in which the 4 guanine that you have here finally fold into an intramolecular G-quartet. And the idea is to use this synthetic quartet or intramolecular quartet to act as a biomimetic G4 ligand, because this approach is called the like approach, because the stability of a G-quadruplex depends on the numbers of its constitutive G-quartet, and by using this synthetic G-quartet, we add another layer of G-quartet, so we obtain a very stable edifice. This was our approach. It was not that simple to demonstrate, but now it's done. But what is really interesting is to keep in mind the fact that we want to use this kind of molecule not for therapeutic purposes. No, we just want to use them as molecular tool to assess the relevance, the existence and functional relevance of G4 within cells. So the idea here is very simple. In fact, the fact that there is a conformational change that is triggered by the G4 ensure a very, very, very high level of selectivity. And I'm really fan about very old-fashioned, quite old-school animation. So everything is represented here. You have the task, and the 4 guanine are here. And the idea is that the 4 guanine self-assemble to form a quartet, and the quartet stabilize the quadruplex, and you obtain a very stable edifice. That being said, till 2011, we developed a series of G4 tasks. I won't enter too much into the detail, but the first example was the biomimetic ligand developed. It is called the DO task, was published with Jean-Louis in 2011. And then we developed the first prototype of smart quadruplex ligand. So the difference is the nature of the guanine. They were neutral. Here there is an amino side chain that can be protonated at physiological pH, and we were able to demonstrate that they do fall within their G4 aphenic conformation only in the presence of the target. This is why we call this ligand the smart ligands. And then we changed the nature of the templates. It was always cycling here just because it's C4 symmetrical. Here we use some pro-fluorogenic templates, naphthalene pyrene, for example. And this was the first example of a twice as smart quadruplex ligand, because they are always smart ligand. They do actually fall into their G4 affinity conformation in the presence of the target only. And the idea is that they were also a smart fluorescent probe, because the fluorescence is triggered upon interaction with that target. And more recently, we developed a series of multivalent tasks, the biotinylated biosite task here, the clickable multitask and azido multitask here, so with a triple bond or with an azido group, and more recently, the photoactivatable and clickable molecule, which is called the photomultitask. So I will select just some example of them, starting with the NAFTA task, because it's quite interesting in terms of photophysics. Again, a wonderful old-fashioned animation. The idea is that, as it is represented here, the template is not fluorescent when the molecule is in its open conformation, but as soon as it interacts with the G4, the intramolecular quartet is formed, and then the fluorescence is triggered. Old-fashioned animation, you see the fluorescence increase upon interaction with the G4 only. So we demonstrate this. It was interesting. So to demonstrate that this molecule is able to interact with the G4, we first use fixed and permeabilized cells, because it's quite convenient to play with fixed cells. So cells were fixed. We then use our NAFTA task. We obtain this kind of images, which was not which was expected, in fact, because we already knew that these very bright foci within the nucleus, which are the nucleoli, were expected to be labeled quite efficiently, and this is the case. But the most interesting thing I highlighted here by white arrows were isolated foci within the cytoplasm. And the interest with playing with fixed cells, we can use a different treatments, for example, the DNS treatment, the RNS treatment, can make some colocalization with the BG4 antibody. And we were able to demonstrate that these foci within the cytoplasm do actually correspond to folded RNA G-quadruplexes. So it was cool, but in fact, a surprising event here is that, okay, it's possible to do it within fixed cells, but their demonstration of the existence of G4 within fixed cells were already demonstrated by Shankar in 2013, so it was not a huge leap. What was interesting with the task is the fact that these molecules are absolutely not toxic. So we can use with live cells. So in that particular case, we just remove the culture medium, add the task, take the image, remove the task, put again the culture media, and then everything goes smoothly. So it was perfectly functional cells, and we were able to spot some foci in the perinuclear region in which the NAFTA task accumulates. So it was the bona fide first demonstration of the existence of G4 RNA within living cells. But the actual point here is that we were forced to play at very high wavelength using a two-photon microscope, which is not routine in the lab. So we were thinking about using this NAFTA task with more classical confocal imaging, but the actual issue was that when you play with the confocal imaging, you use lasers adjusted at a fixed wavelength, usually this one. And clearly, this is a UV-vis of an NAFTA task. It does not absorb light at this wavelength, meaning that it cannot produce a fluorescence at this wavelength. And despite this, we obtained very beautiful images in the three colors. And this led us to discover that NAFTA task is not a classical fluorophore, but especially in cells that are fixed and mounted for the confocal imaging, it behaves as a red edge fluorophore. So the red edge fluorescence is well known for quite a long time, but it was not discovered, or at least not described in a biological medium. So we spent a lot of time to discover this. I won't enter too much into the detail into the red edge mechanism, but if you want to discuss about this, this is very fascinating photophysical, let's say, event. And this red edge fluorescence allows us for developing finally two kind of treatment if we want to use NAFTA for labeling either the DNA or the RNA. So if we use NAFTA in live cells, with NAFTA accumulate, we fix everything, and then we clearly see an accumulation of strong G4 foci within the cytoplasm. So RNA G4s, and if we use this as a post-fixation agent, post-mortem, so we fix everything and just incubate the dead cells with an aftotask, we can see the accumulation of foci within the nuclear compartment, which are bona fide ascribed to G4 DNA. So this simple molecule can be used for detecting DNA or RNA G4, depending on the way you want to use this. And we demonstrate the versatility of this approach more recently, playing not necessarily in MCF7 cells, but in central nervous system cells. In collaboration with Andre, we showed that we can use NAFTA to label G4s in neurons and in astrocytes as well. And this study was interesting because in this study, we demonstrate that the numbers of G4 increase with age of neurons, which means that either G4 are responsible for aging, or aging is more permissive for G4 formation. So it provides a beautiful window of opportunity to investigate whether the stabilization or the destabilization of G4 using small molecule can control aging. And also the versatility of this molecule was demonstrated playing with two different, so let's go back to the cancers. I told you that 80% of cancers are usually considered to be cancers because the telomerase is enzyme, 80%. So the question is, what are the remaining 20%? And the 20% are considered to be ALT plus cells, alternative lengthening of telomeres. So the length of the telomere is maintained, but in a mechanism that is absolutely independent of the telomerase. And using this small molecules and aftertask, we can clearly discriminate in a simple manner in this 2 hours, ALT plus and TER plus cancer cell lines. So it's quite efficient. But we were a bit frustrated by this result because, as it is indicated here, a green dot remains a green dot. I mean, we see something in the cells, in the cell imaging, so that's cool, but how could we be sure that these actually are G4? So to this end, we have to go back to the actual natures of the sequences that are involved. So to this end, we use not the NAFTA task anymore, but this first prototype of bio-T-delated task. And the idea was always the same. It interacts with the G4 with the high selectivity. And then the biotin is used as bait to isolate the complex, the G4 biotask complex. And I'm pretty proud about this old-fashioned animation. The biotin is here and the streptavidin allow for the purifications of the complex, the G4 ligand biotask complex. When we decided to develop this, it was because there was a kind of controversy in the field. The RG4-seq method developed by Shankar in 2016 showed, so it is very simple, you purify your RNA, you split your batches in three, you split your RNA pool in 3 batches, one is untreated, was it treated with a G4 promoting conditions, and then you add a PDS ligand, so it's more than G4 promoting conditions, do some sequencing and analyze in a comparative manner the 3 samples just in order to identify whether G4 fall within this pool of purified RNA. And the conclusion was, from Shankar lab, RG4, RNA G4 is a pervasive secondary structure in the human transcriptome. The same year, David Bartel published in Science another approach, which is based on DMS footprinting, but I won't go too much into the detail. The conclusion of his study that it was that RG4 are mostly enfolded in mammalian cells. So the study finally more or less the same thing, according to 2 different techniques, and the conclusion was completely the opposite. So we reasoned that finally the technique makes a difference. So to solve this conundrum, to be folded or not to be folded, that is a question. We decided to investigate the existence of G4, RNA G4s in vivo. So in vivo in terms of genetics, meaning in fixed chromatins. So the idea was that Finally, G4 can fold, but this folding is certainly very transient. So the first thing that we have to do is to cross-link everything in order to trap the G4 if it is folded in its native state. And then we fragment everything, we use our molecular bait to isolate this and make some sequencing analysis. So this is called the G4 RP-seq for G4 RNA-specific precipitation and sequencing. And this is interesting because it can provide, thanks to this very first step, which is adapted from the ChIP-seq approach, provide with the direct snapshot of folded G4 in vivo conditions. So we perform this, we cross-link everything, we develop this protocol to isolate the RNA, the DNA, to remove the protein, and then we validated this using a qPCR quantification with established G4 forming sequence, and it works quite well. So we move to sequencing stuff. And again, I won't enter too much into the detail, but what we obtain, you have here the 300 ranked transcript analyzed by sequencing, and we analyze the G4 content of this top 100 and less, and the number ranked from 228 to 328. And clearly the G4 content is higher in the more enriched sequences, which means that yes, G4 RNA can be found within cells. And we looked at the sequence and we were quite surprised to see that there were both enriched and downregulated transcript. So protein coding RNA are full of G4s as compared to control, of course, but it was surprising to see that long coding RNA tend to avoid the G4 formation in vivo. So it was quite a surprising observation. The message would be G4 might threaten the functions of long coding RNA. So to gain more insight into this, we decided to incubate our cells with two G4 ligand, BRCA and RHPS4, do the GAR-seq again, and investigate the difference between untreated and treated cells. And again, we obtained that these two molecules, so we selected this because they are well-established telomeric targeting agent, meaning DNA, G4 agent. And I would like to remind you that we are focusing here on RNA. And what we discovered that these two established DNA G4 targeting agents interact very strongly with RNA G4. That's normal because they are highly abundant and more straightforwardly accessible within the cytoplasm. And the common target of this are long non-coding RNA. So the take-home message was this simple approach allows for redefining the mode of action of molecules, and which will provide a source windows of opportunity for different type of therapeutic intervention anyway. So it was cool because we developed this and we showed that using Biotask can help us deciphering the biology of RNA G4. So the conclusion was, yes, G4 RNA can fall within cells. The long non-coding RNA are full of G4, and these are the primary target for G4 ligands. But we next move to the next question is, could we do exactly the same with the not G4 RNA, but G4 DNA? And it was not that simple, but in fact, there are two different techniques that have been developed to study the DNA G4 landscape within cell. The G4-seq method developed by Shankar, and it's in vitro investigation. So he purified the genomic DNA, split it in different samples, treated or not treated, make some sequencing, and the conclusion is that there are more than 700,000 sequences that can hold G4 within our transcript, within our genome, sorry. And the year after, he developed the G4 ChIP-seq method, which is based on the immunoprecipitation of G4 in a chromatin context, which is called the in vivo stuff. And the conclusion was interesting because it showed that roughly 10,000 sequences can be isolated through this approach. We are not at 800,000 sequences, but just 10,000 sequences. And this highlights the suppressive role of heterochromatin for G4 formation within cells. These two approaches are interesting, but they are again, different because they are relying on purified DNA using a G4 ligand or a fixed chromatin using a BG4 antibody. So we asked whether it was possible to do the same using a single molecule, and of course, we want to use our task molecule. So I will go fast because I am really nearly running out of time. Yes. So with this, to investigate this, we use a model genome, a plant genome, a rice genome, because it's really, really convenient to work with the rice genome. And I am involved in a quite long-lasting collaboration with Wenling Zhang in China, in Nanjing, in which he was interested in deciphering the putative roles of G4 and I motifs within rice. So we play with this model, and because a lot of basic work was done, I decided to play with this model to investigate whether we can use the biotask to purify G4s either in vitro or in vivo to make the direct comparison between the two approaches. And interestingly, we also include in our study the antibody BG4. So this was, for me, the first comparison of the capture affinity, the capture efficiency of an antibody on one side, the BG4, and of a small molecule on the other side, the biotask and its new derivative, which is called the biotask, which is now commercially available at Sigma. So we did this experiment, and we were quite happy to see that, in fact, the BG4 and the antibody and the small molecule can precipitate RNA, DNA G4 roughly with the same efficiency. Of course, I won't enter too much into the detail because it's big data science, but we made thousands of different controls. We were even able to isolate roughly 10,000 G4 sequences that were isolated using either the BG4 or the biotask or the biotask. So we can be really confident about the fact that these 10,000 sequences do actually fall the G4 in vitro. The only difference was when we look more carefully to the natures of the G4 that were isolated, the fact that there is a kind of bias of the task toward the exonic G4, while the BG4 is more generic tool for G4s. And in fact, if we look at clearly the fine subtle difference between the two molecular tools, we observe that BG4 is able to enrich more at the 3/4 G4 and the task more the 2/4 G4, but this is roughly the only difference. Of course, these sequences are guanine rich. These are the bias. So I won't enter too much into the detail here, but you can trust me, these molecules are able to isolate G4 DNA as well. And last thing that we did is that we play with the in vitro condition and in vivo conditions and compare this directly this way. And surprisingly or satisfyingly, more than 80% of the G4 sequence that we isolate in vitro and in vivo are common, meaning that this tool can be used in an interchangeable manner in vitro and in vivo. And again, I think that it was interesting because it was the first demonstration, direct comparison of immuno detection versus a chemo detection, or immunoprecipitation versus a chemo precipitation stuff. This is interesting this is my final slide. This is interesting for a point that is of utmost importance. Why we develop these tools? It's not to assess the existence of G4 DNA within cells. So we did this in the case of plants and rice, because it is interesting to identify G4s within this. But what is interesting is the fact that these two techniques, which are called the G4, the GARP-seq and the G4 deep-seq, can be used with or without small molecule treatment. For example, if you incubate your cells with a molecule that is supposed to stabilize G4, DNA on RNA, you use either the GARP-seq or the G4 deep-seq, and you can clearly quantify in an accurate manner the modulations of the G4 landscape, DNA on RNA, using these techniques. And it works quite well. We did this with two molecules, two stabilizers, but We are currently working on a G4 destabilizer. We published last year in the JAX the first prototype of a G4 destabilizer. I'm really excited about. And using this technique, we can see and assess in live, in fact, a modulation of the G4 landscape directly using the G4 DeepSeek or G4 GAP-seq technology. So in conclusion, this design is cool. I mean, we can use the task for fluorescence investigation or optical imaging. We can use the task for purification for many different approaches. And of course, we are many things to do, working with different models and working with our new clickable tool. So 3 of them have been licensed by Merck, and 2 of them are already commercially available at Sigma. So we are currently working on the scope of these 2 molecules, 3 molecules, and of course it is expanded as compared to the biotinylated stuff. So again, to finish, I think I'm a bit right on time. I want to really thank my co-workers, my students, of course, and my long-standing collaborators, my funding agency, and of course you for attending. And I hope that you find this as fascinating as I find it.
Yeah. Thank you very much, David, for this very exciting journey to the research on G-quadruplex. I guess I have many questions, but I guess there are some also in the audience, so we should start first with a question in the audience.
So it's oral question, right?
Yes, at that stage, yes.
Perfect.
So, any question?
Yes, I have one. Oh, well, actually, I have several. But the one I want to ask first is, have you compared, well, first, thank you for your talk, because it was very instructive, I found it. But have you compared the binding of the N task and the bio task to see the difference and how the N task, if the N task was really less specific than the other version of this ligand?
In fact, yes. I did not give you all the details here, but each time we have a new task, because we developed 17 different prototypes, we have a series of in vitro validation, I would say a kind of check. And within these checks, there is affinity measurement, which is we just quantify the KD. And we've always find that all tasks are within the very narrow range of KD, in the micromolar range, between 0.9 and 1.1, roughly speaking, which is logical because, in fact, they do interact with G4 through their quartet, and this is a common feature of all the tasks. Yes. So the question was, are there some influence of the nature of the template for stacking, for example? The answer is certainly yes, but not in terms of affinity and selectivity, because we do not see the difference. And we have recently purchased a BLI machine to better quantify the K on and the K off, so to obtain some more precise information regarding this. And we will screen the 17 different tasks just to assess whether there are small subtle differences that we cannot see using the fluorescence quenching assay, roughly speaking.
Okay, thank you.
Other question? Perhaps I have several. Some general and more focused ones. So I've noticed. So you were speaking about vitro, vivo, and the differentiation. I've noticed that there is on some of your molecules a DOTA. And I was wondering whether you use this also for imaging in vivo, or you were thinking of using it, or there is no point of doing these kind of things?
This was my very first idea in 2011. So you can imagine that it did not work finally. Now, in fact, these are molecular cages. They were selected because they are symmetrical. The symmetry is suited for inserting 4 arms. That's cool. And they are highly water soluble, which is cool because guanine is not really solid. So we counterbalance the hydrophilicity of this molecule using this. Then we tried to use the DOTA, which is not exactly a DOTA, but it's a DOTAM. So this DOTAM, the chelating ability of DOTAM is not that good. So we inserted some endium, some terbium for in vitro validation, and endium, a cold one, just to see whether the metal could stay within the cage, which is not the case. The complex is not stable enough, and in something like 30 minutes, the metal is not in the cage anymore. So it's not stable enough to be used in vivo imaging, small animal this time. But what we are currently working on is to change the nature of this DOTAM to go more towards something which is called the DOT-P, including with the phosphate group, which are better chelating agents for endium, for example, in our case. But the problem is that the chemistry is different because we have to build the arms again. So the DOTA in that case is not useful for chelating, let's say, radiometal, but it can be a problem anyway, because when we do the click, so I did not enter too much into the detail regarding the clickable multitask, for example, but when we do the click, the copper one, the CuAAC, the copper fit within the cyclin quite strongly. So it poisons the activity of the copper, meaning that we are forced to play with 1.1 equivalent of copper. And then we have to remove it from after the click reaction. So it's not stable enough to be used for copper imaging, for example, because this would be the great idea, but it's stable enough to poison, let's say, to complicate the click imaging, the click chemistry.
Okay. Thank you.
Welcome.
Other questions?
Maybe I have another quick one.
Yeah, sure.
It's okay. I was curious about the ChIP-seq workflow for detecting the G-quadruplexes, and the antibody is focusing and is discriminating only the G4 structures over the duplex DNA, or not for it. How is it possible? Is it like there is a specific antigen bound, especially to the G4s?
One of the PI of this study, he belongs to the Shankars Balasubramanian in Cambridge, but his name is David Tannehill. I have the chance to discuss quite a long time this summer with him regarding this, because it's really problematic. The specificity of BG4 is, there is no doubt about the fact that this molecule is incredibly selective for G4s. This has been done by ELISA many times, and it works quite well. But the fact is that we do not know at present what is the part of the G4 that is recognized by the antibody. Meaning that if it's the G quartet, for example, it should not be selective for different type of G4s. We observe in our G4 ChIP-seq experiment that there is a bias using the BG4 toward a certain type of G4s, meaning that it might recognize something else. So it was published the first time in 2013, so 10 years ago, and they fight like hell to obtain some structural detail regarding the way the BG4 interacts with the G4, and finally they failed. And this was rather frustrating. He told me that it's incredibly frustrating. And just what they decided to do is to stop G4 ChIP-seq experiment, because it works quite well, but it's highly prone biased, and you can have many different, it's quite technically challenging to obtain a perfect G4 ChIP-seq data set. And they shift from ChIP-seq to cut and tag, because they told me cut and tag is 1,000 times easier than the G4 ChIP-seq. So now they have invested massive effort in cut and tag and forget about the G4 ChIP-seq, because it's really, really, really complicated to obtain. And unfortunately, in our case, I discussed with my colleague in China and said, maybe G4 ChIP-seq is cool, but we can play with cut and tag, and it does not work in our eyes. So we do not know exactly why. So we have solved an issue, stop playing with G4 ChIP-seq because it's really, really time consuming, but we have now a new challenge to address, because cut and tag does not work quite well. And just to complete my answer, a couple of weeks ago, Shankar published another technique, which is not the cat and tag anymore, but which is called the ChemMap, in which he forget about using the antibody and play only with small molecule. And the small molecules are biotinylated, which means that biotask is suited to be using ChemMap. So that's cool. And just the idea is that PG4, just like every antibody, is a beautiful molecular tool, but it's very difficult to produce and to purify, which is the bottleneck. And this is what motivated us at the very beginning to try to find a small molecule that is easier to at least synthesize and characterize and so on, because it's far easier to play with a fully characterized molecule rather than a quite complicated antibody. But it works quite well. This is why we are quite happy about the G4 G6 stuff, because we have the direct comparison. We claim that we can use in an interchangeable manner antibody or G4, but antibody or small molecule. But the question was always the same. Could you just demonstrate it? No, that's done. It 2 years of work, but it's done right now.
Okay, thank you
very much.
So I don't know if there are other questions in the audience. If not, then we have our flash presentation, and actually Matthieu is our presenter this morning. So perhaps, yeah, very good.
Okay. Can you see my presentation full screen? Is it fine?
Yes, this is perfect.
So thank you. Good morning, everyone. My name is Matthieu Ranz. I'm a second-year PhD student in the ARNA laboratory. And today I will introduce a new technique for the study of G4 DNA structures. So the first thing to consider is that G-quadruplexes are highly polymorphic structure. And depending on several parameters, such as the sequence, the cation, or small molecules, they can adopt multiple conformations in equilibrium with different number of tetrads or different loop arrangements. So to illustrate it, I use the telomeric sequence formed by 23 TAG that falls into 3 conformers in presence of potassium ions. Especially, it forms a 2-tetrad G-quadruplex, so with only 1 potassium bound in between the tetrads, and 3 tetrads G-quadruplexes with 2 potassium between the tetrads. So this latter, namely hybrid 1 and hybrid 2, differ only by their loops geometry. So this kind of conformational ensemble is quite difficult to analyze with usual techniques. So that's why our goal is to develop new methods to characterize such ensembles using native mass spectrometry. And in native MS, we can characterize well the G-force behavior in the gas phase. Indeed, in a mass spectrum, we observe different peaks corresponding to the DNA with different stoichiometries of potassium bound. The 0K plus so corresponds to the unfolded form, or the 1K plus corresponds to the 2 tetrads quadruplex, and so on, and the 2K plus to the 3 tetrads quadruplex. And in the case of 23 TAG, remember that there is this 2 hybrid structure that coexist here. So to learn more in details about the structural dynamics in solution, we couple an isotopic labeling method that provides additional structural information. And this technique is hydrogène-deutérium exchange. And this isotopic exchange, it occurs at a rate depending on both the involvement of the protons in H bonding and the solvent accessibility. So you can note that in G-quadruplexes, the exchangeable protons, the amino protons and the imino protons are well protected inside the tetrad. So what we do to trigger STX, we incubate a deuterated DNA sample in water. So it's actually a D to H exchange that is inducing a mass decrease of the structure. So this is effectively what we observe by mass spectrometry, a gradual shift of the mass envelopes. And at every incubation time point, we can measure the mass of each species. So we convert this mass into a number of sites. Excuse me. And we plot it as a function of the time. So this way we have, for every species, a kinetics profile that matches the number of tetrads in solution and therefore reflects the structure. However, for the 23TAG sequence, these yellow curves represent the average kinetics of these two conformers. And with this technique in itself, it's not sufficient to determine individual profiles for each of the conformers of same mass. So to overcome this challenge, we coupled our method to ion mobility mass spectrometry. And indeed, in the ion mobility mass spectrometers, ions of same mass but different shape, here represented in green and in blue, can travel through a drift tube filled with gas. And the more compact ions will travel through the tube faster because they make fewer collisions with the gas. So this provides a separation by shape of conformers of same mass, and practically it generates what we call an arrival time distribution, where each peak corresponds to one conformer. In the case of the sequence 23TAG, this is what we obtain: a map with the x-axis corresponding to the M/Z separation, and the y-axis corresponding to the IMS separation, as previously explained. So now we select the peak containing the conformers of interest, and we observe two ion mobility populations. We then select appropriate zones, and we extract the corresponding mass spectra of these different zones, so of the two different populations for every time point. And this is what we observe. So although the exchange is quite similar for the two species, because the two structures are not so different actually, we observe still significantly different exchange rate in green and in red. That proves that the two conformers detected in the gas phase really exist in solution, and that we were able to measure kinetics of exchange for the two conformers of same mass with this method. And that was kind of the goal. So all in all, we are confident that this technique can help to characterize such conformer mixtures. And in conclusion, we can say that we managed to, in a single experiment, and this is important, to disentangle, to couple three techniques, the HDX, the native MS, and the IMS, to disentangle polymorphic mixture of biological interest, such as the 23TAG presented today. And well, we think that this technique kind of opens new avenues to decipher all others G4 conformational ensembles. And currently, we are applying the same workflow to the study of G4 ligand complexes. So yeah, with this, I'd like to thank the STS department for this opportunity today, and the ANR for funding, and of course my team at ICB, especially my supervisor Eric Largy, Valérie Gabellica, our group leader, and Frédéric Rosu for the mass spec facility. Thank you for your attention.
Thank you, Matthieu, for this very interesting talk. I don't know if there are any questions related to the presentation from Matthieu.
Yes, may I?
Yeah, sure, of course.
Cool. Regarding just your previous slide, not this one, but the previous one, exactly.
This one? Mm-hmm.
Yeah. The difference is significant between the, let's say, green and red curves as compared to the yellow one.
Yes.
Did you try, so the information that is missing here is reproducibility of this, because it can be small difference, but significant in terms of statistics. So did you perform these experiments many different times, and did you see some statistically different?
Well, the critical part of this result is the selection of the areas in the ion mobility.
Mm-hmm.
So what I did is I selected different zones. So this one is quite big, but I've tried like bigger selection for the red zone, for example, and also smaller. And the idea is trying to not overlap with the second species that's sitting behind, because this generates like a bias. So we need enough signal to have to be able to characterize it, but not so much so we can overlap. So I've tried different selection of the areas, and the curves are matching quite well. I can even show you the results because I think I have this in backup. There we go. For the 23TAG, I've selected like 4 zones in this range and in this whole range, And you can see that the difference is still significant for all the possible selections. And for comparison, here you have different other sequences that have different ion mobility, but only one structure no, and we see that they perfectly matches to one another. So this is not a systematic bias due to the selection.
Okay, so this is reliable,
yes.
Short but reliable. And do you try to exploit it in a quantitative manner? For example, the kind of IC50, KD, whatever, because it would be a constant in minutes. So did you try to exploit this?
Yes, actually, after further deconvolutions of the mass spectra that we obtain, we can observe actually two different regimes. And maybe I can also show this slide. No, sorry. It's in a certain way, you can access the constant of opening of, there we go. You can access two different regimes depending on the way these species exchange. There is, for not so stable structures, such as the 23TAG sequence that I've shown, a part of the structure that is partially unfolding. This way, the constant that we measure with this rate of exchange can simplify into just the opening constant, which is which become the limiting event. So this way, by selecting only the contribution of the species that partially unfold and then refold, we can access this rate of exchange, which is this rate of opening, which is quite consistent.
Mm-hmm. Mm-hmm. That's interesting. And when you play with ligand finally, did you see some difference in drift time?
Actually, we are just starting the experiments now with ligand, and we didn't have yet the opportunity to couple the technique with ion mobility mass spectrometry. So for now, it's just focus on native MS and of ligand binding. So it will be like more the quantitative exchange without the ion mobility separation.
Mm-hmm. But it's fascinating because you have access to another level of detail regarding the interaction of the small molecule with the G4 ligand. So thank you so much. It's a great job.
You're welcome.
Absolutely. Thank you. Is there any additional question to Matthieu? So if not, the point of the organization is that Matthieu can now share a private moment with David. So I don't know, Valérie, Valérie Deverne, you have you are the animateur de la session, so maybe you can organize this.
Je crois que Matthieu est animateur maintenant.
Okay, so so then everybody can, of course, leave the room. So I would like to thank David again for this very nice presentation, as well as Matthieu for this very good seminar. And so thank you all for attending it. And I would like to give you the new date again. So it's June 29 for the presentation by Jean-Christophe Leroux for this department webinar series. So thank you very much. Thank you, David, and see you soon.